Okay, guys, last chapter for the test on Wednesday. I will be reviewing you guys on Tuesday. I sent you guys a text message earlier today. Um, the last chapter focuses more on the origins of eukaryotic diversity. We already talked about prokaryotes in chapter 27. So the first of the eukaryotes are going to be simple, unicellular, macroscopic um, organisms that arise about 2.7 billion years ago, and they're called protists collectively. Now, the kingdom Protista has been under heavy remodeling because of all the modern systemists today. They want to check for. They're using new methods in order to reclassify them into different types of kingdoms. Now, I know you guys have probably heard that protists are all eukaryotic organisms that don't fit into fungi, animalia, or plantae. So it's problematic because they do contain mainly unicellular eukaryotes, but it also contains any organism that is not classified as a plant, a fungus, or an animal. So the kingdom then is paraphyletic, and if you remember back, paraphyletic means that the members of each clade fit into more than one different clade and includes some but not all of the ancestors' descendants, which violates the rules of what a clade is. So it's since been considered to be split into 20 plus kingdoms, but it is still used, protist is still used as a general term for the entire organism group. They are the most diverse of all the eukaryotes. They vary in structure and function. Most of them are unicellular. They're the simplest, but they are a lot more complex than you would think on the cellular level, meaning that they do everything that multicellular organisms do, but they do it as a single cell. So in terms of nutrition, let's see, on figure 28.3, that's what they're looking at here. Most are aerobic, some are anaerobic, some are respiring bacteria. Most of them are photoautotrophic with chloroplasts. Some of them are heterotrophic by absorption or ingestion. Some of them are both, when they call those mixotrophs. And here is an example, a euglena, right here. Now, when I describe the characteristics of protists, I'm going to be describing all types of characteristics. That's the problem with protists, is that they don't fit into just one category and have a specific amount of characteristics that are unique to their clade. So, they are diverse. Nutrition isn't really, as you can see, it's not a very good, reliable taxonomic characteristic because they fit into all three types. They have characteristics of all the other three eukaryotic kingdoms, meaning that protists are going to be the first that evolve, the first eukaryotes, because each kingdom, plantae, fungi, and animalia, have characteristics that fit into all three protists. That fit into the Protista kingdom. So they can divide them ecologically then, the protists, into three types. All right, you have the animal-like protists, which are called protozoa, and they mainly function in ingestion. Fungus-like absorptive protists that mainly function in absorption. And then you have the photosynthetic protists, or the plant-like protists, that would be all of the ones that are photoautotrophic. In terms of motility, most of them have flagella or cilia for motility. Flagella are more extensive in eukaryotes than in prokaryotes, but they are not homologous. Okay, so the flagella that arise in eukaryotes are not the same as the flagella in prokaryotes. That's important to remember. When we look at their life cycles, are going to be varied as normal. Usually include some stage or phase. The phase is similar to mitosis. Some are asexual only. Some are sexual only with meiosis and syngamy. Most produce resistant cells called cysts at some point in their life. Okay, it kind of mirrors the endospore theory of prokaryotes. It has the same type of function. Their habitats are mostly aquatic, all right, anywhere where there is water. They form a lot of what we call phytoplankton, which is the base of most marine food webs. They can be free-living and pathogenic, so pretty much they have the characteristics of all eukaryotes. Now, when we look at their origin and early diversification, we're going to be talking a lot about the endosymbiosis theory. Okay, so here in figure 4.28.4, you're looking at symbiotic relationships between earlier prokaryotes. All right, so you guys all should know the theory behind in the endosymbiotic theory, right, where one prokaryote engulfs another, and instead of digesting it, it actually uses it, and they form some sort of mutualistic relationship. And then therein lies the origins of the eukaryote. So the small genome of prokaryotes limits the number of genes that are available from metabolic activities. So it involves a lot of steps in evolution to this increasing complexity. All right, first you have to have multicellularity, which include most of the filamentous cyanobacteria. Then they have to develop symbiotic relationships, and then they have to be able to compartmentalize their different metabolic functions. But how? How do they do this? Well, for multicellularity, they have a theory that maybe the endomembrane system evolved from specialized infoldings of prokaryotic membrane. So in this case, right here, you're saying that the plasma membrane actually folds in on itself and then creates this endomembrane system. For symbiotic relationships, it's talking more about endosymbiosis, where it starts to engulf some free-living prokaryotes that have specialized functions. Say they can make ATP, as in mitochondria, or they can harness the energy of the sun, as in chloroplasts or plastids, is what they're collectively called. This is how we explain the evolution of mitochondria and chloroplasts. And we're going to go into it in a little bit more detail in a minute. But it fits the idea of Darwinian refinement of existing structures. All right, so we're not exactly creating this new eukaryote. It's actually just a remodeling of previous structures over a period of time. So they give rise to what we call eukaryotic organelles. So the organelles used to be free-living prokaryotes, and now they have formed some sort of mutualistic symbiotic relationships that is now irreversible, meaning that if they were to go back, they cannot live on their own by themselves, and we're going to talk about it in a little bit. So they believe that the origin of eukaryotes doesn't mean that it was just one endosymbiotic, event, that, this one huge big prokaryote just engulfs all of its organelles or the prokaryotes at one time. So they think of it as serial endosymbiosis, meaning that a small prokaryote living in larger host cells will be engulfed many times, right? That's why they believe this is true, because mitochondria and chloroplasts have a double membrane system. So every time that there is a membrane, it signifies an endosymbiotic event. So the mitochondria means that ancestors were aerobic heterotrophic prokaryotes that became endosymbionts. And for chloroplasts, it means that they were photosynthetic prokaryotes that became endosymbionts. But how? How do they do this? Either it enters as an undigested prey or an internal parasite. If so, then the endomembrane and cytoplasm must have evolved first to be able to engulf and package these prokaryotes in little vesicles, right, as what normal endosymbiosis is. These heterotrophic hosts use nutrients by the photosynthetic endosymbionts, and in an increasing aerobic environment with the oxygen accumulating in the atmosphere, anaerobic hosts use aerobic endosymbionts in their quest to survive. So eventually, the prokaryotes are going to, over time, become very, very interdependent.